Hello, my name is Shub Thacker, and over the past summers, I've been investigating a mechanism of our microbiota that can aid in our fight against the Clostridium difficile infection epidemic. But first, what is Clostridium difficile infection, or C. diff? Sprouting from the toxins of the Clostridium difficile bacteria, it is the leading cause of nosocomial diarrhea and thus is a great risk for hospitalization because a majority of cases occur in healthcare systems such as hospitals or nursing homes. According to the CDC, C. diff infections infect about 500,000 Americans a year, with unfortunately 30,000 of them dying due to the disease itself or related complications such as rampant diarrhea, intestinal epithelium degradation, or toxic megacolon. The most common treatment for CDI is through oral antibiotics, such as vancomycin and metronidazole. However, as Winston and Theriot and other reviews highlight, the influx of antibiotics may actually be detrimental to patients' health and may progress pathogenic growth because it can lead to a striking recurrent infection rate up to 20 to 30%. So why does this occur? Literature explains that the pathogen best grows in states of gut dysbiosis, which is when the strata of bacteria in our gut is decimated through means such as antibiotics. This is noteworthy because 1. The pathogen itself is very antibiotic resistant, so it may linger after the administration of antibiotics. Number 2 is that the gut microbiota plays a critical role in inhibiting pathogenic growth. One such checking mechanism is the production of secondary bile acids supplemented by enzymes produced by Clostridium scindens and Hieronymus. Primary bile acids produced by the hepatocytes of cholic acid and chenodeoxycholic acid and their glycine or taurine conjugate counterparts are co-germinants of the pathogen, as Stork 2016 found there is a 10 to the 5th fold increase in pathogen recovery rate in vitro experiments when pathogens are cultured with taurocolic acid and cholic acid. However, Kang et al. And other studies cite the inhibition of pathogenic growth through the production of secondary bile acids of lethocolic acid and deoxycholic acid, which is mediated by the process of 7-alpha dehydroxylation. This process is controlled by a few species of the Clostridium and Eubacterium genera, with the bile acid inducible or BAI operon. The conversion from primary bile acid to secondary bile acid is mediated by 7-alpha dehydratase, coded by BAIE, whose step is irreversible. Studies have found that this gene is highly active in Clostridium scindens and Hieronymus. Now remember the state of gut dysbiosis in patients because of the influx of antibiotics? Well, these beneficial bacteria are now gone. So to replenish these pathogen-inhibiting bacteria, fecal microbiota transplantation, or FMT, is gaining legitimacy to treat C. diff infections per mins et al. Based on previous knowledge, the reduction of primary bile acid abundances and increased secondary bile acid abundances induce inhibition of CDI. Thus, I hypothesize that levels of secondary bile acids increase and abundances of primary bile acids decrease from a pre-transplant state to a post-transplant state, as BAIE is expressed more post-transplant. For my experiment, samples were chosen based on their bile acid detectability and infection status. In most FMT cases, there are 4 groups of feces involved: infected pre-transplant patients, patients at 1 week post-transplant, patients at 3 month post-transplant, and a healthy donor. For 8 cases, I helped to conduct HPLC-MS to quantify the abundances of primary and secondary bile acids. And next, I conducted PCR to detect the BAIE gene of both species throughout the FMT trial for 18 cases using primers I designed in silico. So let's look at what I found. If we look at figure 4, there were notable differences in bile acid levels between donors and pre-transplant recipients. Generally, the infected patients had markedly higher levels of primary bile acids and had very minimal amounts of secondary bile acids compared to the healthy donors. Out of the 6 primary bile acids, only cholic acid and taurochenodioxycholic acid were tested to have a significant difference between the two. Also noteworthy is a significant discrepancy in the levels of secondary bile acids between the 2 groups, with healthy donors exhibiting higher levels. Figure 5 and 6 display abundance data throughout the typical FMT case with 3 stages: the infected pre-transplant patients, 1 week post patients, 3 month post patients. If we notice figure 5, primary bile acids typically do not significantly change after the procedure, though there are some marked decreases. Only cholic acid, taurocolic acid, and taurochenodioxycholic acid were the acids whose abundances decreased significantly, at 13-fold, 25-fold, and 24-fold respectively. This is extremely important because this suggests that the overall abundances of co-germinants of C. diff significantly decreased after the transplant, supporting my hypothesis. Also noteworthy is that the most stark changes occurred after 1 week, and these levels were relatively maintained after 3 months. Let's look at the figure 6 now. Secondary bile acid abundances significantly increased after the FMT. From the pre-transplant state to the 1 week post-transplant state, there were significant average increases of lethocolic acid and deoxycholic acid at 408-fold and 84-fold. After 3 months, like the primary bile acids, these levels were relatively maintained after their significant spikes 1 week after the transplant. It must be noted that after 3 months, all the patients were healthy. This suggests that the significant increase of bile acids, particularly the secondary bile acids, after the transplant is critical in inhibiting pathogenic growth, thus supporting my hypothesis. Now for a holistic look into the ideal bile acid pool composition, Figure 7 represents the average composition of each stage of the FMT procedure. As we can see, there are tiny portions that represent the secondary bile acids, but as we move forward, the compositions start to be heavily secondary bile acid concentrated, around 90% for the 1 week post, 3 month post, and the healthy donor. It also must be noted that the latter 3 stages included all healthy patients and donors. Most importantly, the healthy donors and post-transplant patients had similar bile acid pool compositions, thus suggesting that a heavy abundance of the secondary bile acids and a minimal abundance of primary bile acids is important and optimal to be healthy and reduce pathogenic growth. So let's move on to the genetic aspect of my project. Figure 10 shows the samples with detected C. syndens BIE gene in 18 cases, so let's establish a baseline. The healthy donor group had gene presence that was significantly higher than the infected pre-transplant group. Similarly, differences in Clostridium sindens BIE presence is significant between the pre-transplant and 1 week post-transplant groups. Majority of these cases saw either an increase or a constant in gene detection. Notably, this 1 week post profile also matched a majority of 3 month post and donor profiles because there was no significant difference between the three. Figure 11 shows samples with detected C. hiranonis BIE gene in 18 different cases. However, the results were starkly different from the results of C. syndens BIE. There was no significant difference in detection of C. hiranonis BIE gene presence between the donor and pre-transplant samples and the pre-transplant and 1 week post-transplant samples. So the genetic aspect of my project suggests that there's an increase in gene presence of C. syndens BIE and thus possibly an increase in 7-alpha dehydratase production from the species. However, this is not the case with C. hiranonis. This study sought to investigate the possible mechanisms of fecal microbiota transplantation's high successful treatment rate of C. diff by measuring the differences of primary and secondary bile acids and the bacterial gene BIE that is responsible for secondary bile acid production before and after the transplant. Primary bile acids, germinates of C. difficile pathogens, reduce drastically, and secondary bile acids, which are the inhibitors of the pathogens, increase significantly after transplantation. And furthermore, my genetic aspect sought that the BIE gene of C. syndens had a significant increase in detection post-treatment, but further exploration through quantifying protein expression will further develop the BIE's role in the compositional change of the bile acid pool, and subsequently the decrease of C. difficile pathogens post-FMT. Hopefully, this new data that can be generated from this protein quantification, along with my own data, will then serve as a possible avenue to develop therapeutics like drugs to help treat the C. difficile epidemic in the United States and in the world. Thank you.